classes. Live classes are to elevate the learning of the student, in which the student will have the direct interaction with the faculty. Then also on Auricle, all the student will have the access of the recorded sessions also. So if any student have missed any the class, so of any subject, so the student can have the class anytime, anywhere through the recorded sessions. And also we provide notes with CBME based question answers, MCQ, and full zone, full self-assessments also there. Then also MCQs are there from beginner level to expert level, which contain standard multiple choice questions. Then also student can book a slot for personalized counseling session, in which the student will have the direct interaction with the counselor. Then on Auricle, we provide some subscription plans starting from 3 months to 12 months. So basic plan is of 3 months, standard plan is of 6 months, and premium plan is of 12 months. The premium plan includes all the services that is provided by Auricle, that contains like pre-recorded class, live doubt clarification, one-to-one personalized guidance, notes, quick revision, examination module. And this premium plan is cost around 50,000. And also for instant 20% discount, you can use my code Anchal20. So I would recommend all the students to take this premium plan of 12 months. Then as we have started the internal marathon exam series from April 22, so the features are like rapid revision of all the important questions of all the units. And also we discuss about the volatile fact for quick revision, and multiple choice questions also we're going to discuss. And also we assure top note grades in exam. And this marathon series is beneficial for all the student of any university exam of all the states also. So these are the important topics that we're going to cover in today's session, that is cerebellum and hypothalamus receptors and its classification and properties also we're going to discuss. Then we're going to discuss about front lobe and parietal lobe syndrome, then EPSP and IPSP we're going to discuss, then invert stretch reflex we're going to discuss, and decerebrate rigidity we're going to discuss in today's session. So what is receptor? So receptor is a protein molecule, right? That is usually embedded within the plasma membrane surface of a cell that receive chemical signal from outside the cell. When such chemical signal bind to a receptor, and they cause some form of cellular tissue response. As we can see, the receptors are there over which the protein molecule is going to bound, and then changes will be there. Okay, so in another way we can define the receptors are specialized sensory afferent nerve ending that give rise to sensory stimuli, right? And these sensory nerve ending terminates in periphery as bare unmyelinated endings, or in the form of specialized capsulated structures, right? So when stimulated, receptors produce a series of impulses that are transmitted through the afferent nerves, right? So now we're going to discuss about the screen is not visible.
Yes, ma'am. Screen is not visible. White screen I am able to see. Some MS Word screen. Actual screen is not there.
Now?
Yes, ma'am. Now okay.
Okay, so receptors are there. So in receptors, we classified it as exoreceptors and interoreceptors. So as we can see in the picture also, in the slide, the exoreceptors includes chemoreceptors, mechanoreceptors, photoreceptors, thermoreceptors, and nociceptors. Okay, so what are the chemoreceptors? Chemoreceptor means, as the name suggests, which detect any kind of change in a chemical substance to generate the biological signal. And these are also for taste, smell, blood, in blood, they basically detect for the constituents like CO2, O2, glucose, amino acids, and fat. Okay, and these receptors are basically present in hypothalamus. Okay, so this is all about chemoreceptors. And please remember, here we are only discussing about the exeroreceptors, not interoreceptors. So next we have mechanoreceptors. So mechanoreceptors are those receptors which relay extracellular stimulus to intracellular signal transduction, and they detect stimuli from touch, pressure, hearing, and equilibrium. And also free nerve ending, expanding, tip ending, and stray ending, right? Then next we have photoreceptors. So photo, as the name suggests, photo means light, right? So they produce response to stimuli and light, and basically the major types are rods and cones cells in the eye. Okay, then next we have thermoreceptor. Thermo means temperature, right? So they're going to detect the heat, energy, temperature in the environment and in the body, and also they are free nerve endings. Then nociceptors means the receptors which are present for pain, right? So here they detect signal from damaged tissue or detect sensation of pain, and free bare nerve endings are found in particularly skin, muscles, joints, and bones. So these are all about the exeroreceptors. And what are exeroreceptors? Exeroreceptors are those receptors which give response to stimuli from arising from outside the body. Okay, so please remember that the exeroreceptors are the receptors which give response to stimuli arising from the outside. Okay, and also if the question over the receptor will come, and there are highly probability that because the receptors are the most important topic of the CNS, so the question will come like that, what are receptors, define receptors, classify receptors, and discuss all the important properties of receptors. So this is how the question can come. And also whenever the question will come on the receptor, so highly chances are there that the question will come in long answer. So next we have interoreceptors. So interoreceptors, as the name suggests, intero means within the body. Okay, so these are the receptors which detect or which responds to stimuli arising within the body, right? And also the interoreceptors are of two types. The first we have visceroceptors, then second we have proprioceptors, right? So what is visceroceptors? Visceroceptors means that are situated in the viscera. Okay, so that's why they are known as visceroceptors. And proprioceptors, as the name suggests, proprio means these are the receptors which give response to the change in the position. Okay, so please remember, these are the receptors which detect change or which give response to change in position of different part of the body. Okay, then as we have discussed about viscera, so they are situated in visceral part. Right. Next, we have the properties. So here we're going to discuss the properties of receptors, which is also an important topic, and also the question asked in the university exams also about the properties. So about the properties of receptors, first we have specificity of response, and that is defined by the Muller's law. Okay, so specificity of response refers to the response given by a particular type of receptors to a specific sensation. Okay, so if in example we take like for example, pain receptors give response to a pain sensation only, right? Similarly, temperature receptors give response to the change in temperature. Okay, and also each type of sensation depend upon the part of the brain in which its fibers terminate. Okay, so this is all about specificity of response, means the particular type of receptors only responsible to detect a change in a particular thing, right? Now next we have the another properties, that is adaptation. adaptation. So here we're going to talk about the sensory adaptation. Right? So adaptation is the decline of discharge of sensory input, basically is the decline in the discharge of sensory impulse when the receptors is stimulated continuously with constant strength, right? That's why it is also known as sensory adaptation, or it is also known as desensitization, right? So here we are only talking about the sensory receptors. So in sensory adaptation, we have two types of receptors. The first we have phasic receptors, and second we have tonic receptors. Okay, so phasic, basically depending upon the adaptation time, receptors are divided as phasic receptors and tonic receptors. So first we're going to talk about phasic receptors. So phasic receptors are those receptors which get adapt rapidly. Okay, for example, touch and pressure receptors are the phasic receptors. So according to any kind of touch, any kind of pressure, the body will adapt rapidly. Okay, next we have a tonic receptors. So tonic receptors are those receptors which adapt slowly. Okay, so here also I'm going to write the phasic receptors which adapt rapidly, and the tonic receptors which adapt slowly. Okay, so in tonic receptors, muscle, spindle, pain receptors, cold receptors are the type of tonic receptors which adapt very slowly. Okay, next the another property we have response to increase in strength of stimulus. Okay, and here it is defined by Weber-Fechner's law. Okay, so basically what happened during the stimulation of the receptors, if the response given by the receptors to be doubled, right, then the strength of the stimulus must be increased by 100 times. Okay, and this phenomena is stated by the scientists, the Weber-Fechner law, and which state that the intensity of response means the sensation of a receptors is directly proportional to the logarithm increases in the intensity of stimulus. Now, next property we have their sensory transduction. Okay, what is sensory transduction? Sensory transduction is in a receptor is a process, right, by which the energy, or we can call the stimulus in the environment, is converted to the electrical impulse. Electrical means the stimulus is converting into the action potential, okay, in the nerve fiber, right? So transaction always means conversion of the energy from one type to the another. That's why it is also known as sensory transduction, and this is the property, this is the another property of the receptor. Okay, so whenever a receptor is stimulated, it gives response by sending information about the stimulus to the CNS, and series of events that occur to carry out these functions, such as the development of receptor potential in the receptor cells and the development of action potential in the sensory nerve. Okay, so example, just now we have discussed, and another example could be the example of chemoreceptors which convert chemical energy into the action potential in the sensory nerve fiber. Okay, and also the touch receptors which convert mechanical energy into the action potential again in the sensory nerve fiber. Okay, then another property we have of the receptor potential. Okay. What is receptor potential? Is a non-propagated transmembrane potential difference that develops when a receptor is stimulated, and that's why it is also known as generator receptor. Okay, and the receptor potential is of short-lived, and hence this is also known as transient receptor potential. Now, receptor potential is not not an action potential, right? It is graded potential, and it is similar to the excitatory postsynaptic potential, EPSP, that also we're going to discuss in today's class. What is EPSP? So this is similar to EPSP in synapse and unplayed potential in neurotransmitter junction, and electrotonic potential in the nerve fibers also. Then another property of this receptor we have, and the last one we have, law of projection. So what is law of projection? Whenever a sensory pathway from receptor to cerebral cortex, right? So whenever any pathway, whenever any sensory pathway from receptor to cerebral cortex is gets stimulated on any particular site along its course, then the sensation caused by stimulus is always felt at the location of receptors, irrespective of site stimulated, and also this phenomena is known as law of projection. Okay, so these all are the properties, are very important properties of the receptors. And also if the question over the properties will come, so you have to state all these properties of the receptors, and along with the properties, along with the name of the properties, you also have to describe the phenomena, right? Next, we're going to learn about one of the mechanism that also should be described in the property of the receptors, that is development of receptor potential, how it works. Okay, so as we know, receptor potential causes the development of a local circuit of current flow, right? So when spread along the unmyelinated part of nerve fiber within the cell, so when this local circuit of current reaches the first node of Renvier within the cell, so it causes the opening of the voltage gated sodium channels, and there will be influx of sodium, means there will be entrance of sodium into the nerve fibers, and this lead to the development of action potential in the nerve. So this development also we're going to learn with the help of a flow chart. Okay, so what happens as there will be pressure stimulus, so this will lead to compression of Pacinian corpuscle, right? Then this will lead to elongation or change in shape of Pacinian corpuscle. Then it will lead to deformation of center core fiber, and there will be opening of sodium channels. Okay, and influx of sodium will be there, which leads to development of receptor potential. And as the development of receptor potential will be there, it will lead to development of local circuit of current flow, right? And again, there will be opening of voltage gated sodium channels. Again, entrance of sodium ions will be there, which leads to generation of action potential in the nerve fiber. So student, the question over the development of receptor potential can also come differently in a short note. So if that question will come, so you have to mention the entire flow chart. That is also very important. And also if the question over the receptors will come in the long answer, then also you need to explain the entire development of receptor potential under the heading of receptor potential. Okay. So this is all about the receptors. Next, we have the another topic that just we were discussing about EPSP, right? EPSP and IPSP that we're going to discuss. So in the last class also in the function of synapse, we discussed that there is excitatory synapse as well as there is inhibitory synapse. Okay. So what is EPSP? EPSP is excitatory post synaptic potential. Okay. And what is IPSP? It is inhibitory post synaptic potential. Okay. And the picture which we can see here. So this is all about the summation that we're going to discuss in the end of this topic. Okay. So first we're going to talk about EPSP. So what is EPSP? EPSP is the non-propagated electrical potential that develop during the process of synaptic transmission. Okay, so whenever any action potential reaches the presynaptic action potential axon terminal, that we have seen in the last class also, that how this whole process can occur. But if you have not watched that series, so you can see that class in the recorded sessions. So the voltage gated calcium channels at the presynaptic membrane will get open and there will be influx of calcium channels. Now, so the calcium channels causes the release of neurotransmitters, which substances which cause the means of exocytosis, right? That we also discussed. So the neurotransmitter which is excitatory in function, that is known as excitatory neurotransmitters. So here the important role occur by the excitatory neurotransmitter. So neurotransmitter. And here for the neurotransmitter, the example, the most popular important example we have acetylcholine. Okay. Now neurotransmitter which are excitatory in function passes through the presynaptic membrane and will there in the synaptic cleft. We have discussed in the last class what is synaptic cleft, that reaches the postsynaptic membrane, and now the neurotransmitter bind with the receptor protein present in the postsynaptic membrane to form neurotransmitter receptor complex. Okay. Now, neurotransmitter receptor complex causes the production of a non-propagated EPSP and commonly excitatory neurotransmitter that already we have discussed, that is acetylcholine, right? Then also when we talk about the properties of EPSP, so talking about the properties, the properties we have is that EPSP is always non-propagated and it does not obey all or none law. Okay. Then next we have IPSP. So as the name suggests, IPSP means inhibitory postsynaptic potential, and here we have the inhibitory neurotransmitters. An example, the most familiar example is GABA. Okay. So, postsynaptic inhibition is a type of synaptic inhibition that occurs due to release of inhibitory neurotransmitter. So, please remember here we will have the role of excitatory neurotransmitter in IPSP we have the role of inhibitory neurotransmitters. Okay. So, from presynaptic terminal instead of the excitatory neurotransmitter substance. So, that is why it is also known as direct inhibition, and the inhibitory neurotransmitters are GABA, dopamine is also there, glycine is also there. Okay. So this is all about the IPSP. Then also, we're going to discuss about the summation. So what is summation? First, we should know what is summation. Summation is in the synapse is a fusion or aggregation of familiar impulse. Okay. So it is a fusion or aggregation. Means this will be the aggregation of EPSP plus IPSP. So together EPSP and IPSP is going to determine whether the action potential is going to develop or not. So that's why the summation is known as the fusion or aggregation of many impulse or stimuli. Okay, and in another word we can also call that it is a fusion or effect of progressive increase in the excitatory postsynaptic potential in postsynaptic neuron. Okay, normally one single EPSP is known as graded potential that has a very little effect on postsynaptic membrane potential, so it cannot reach the threshold limit to produce an action potential. Okay, when many presynaptic excitatory terminals are stimulated simultaneously, then many EPSP will develop simultaneously at a time, or when single presynaptic terminal is stimulated repeatedly, many EPSP will be there. That is enough to generate the action potential. Okay, now here we're going to learn about the spatial summation and temporal summation. Okay, now what is spatial summation? When an action potential is produced by the fusion of many EPSP, right? Development from many presynaptic terminal, that time it is known as spatial. Okay, so here what is spatial? Fusion of EPSPs, okay, developed from many presynaptic terminals. Okay, and summation of all these EPSP allow the potential to reach the threshold level for generation of an action potential in the postsynaptic neuron. Okay, and whereas the summation of IPSP prevent the generation of action potential. Okay, so always remember the summation of IPSP is always present to prevent the generation of action potential. That's why it is also known as inhibitory, because it's inhibit action potential. Okay, now next, we have temporal. Temporal, now what happens in temporal? When the action potential is produced by fusion of many EPSP from a single presynaptic terminal. Okay, so EPSP from single presynaptic terminal. That is known as temporal summation. Okay, and here EPSP develop one after another. Okay, and all the EPSP overlap and summate with each other so that the action potential can be generated in the postsynaptic neuron. Okay, so temporal summation occur when one presynaptic terminal is stimulated repeatedly, and both spatial and temporal summation play a very important role in the facilitation of the response. So this is all about EPSP, IPSP, and the summation. Okay, then next we have the another and the very important topic of CNS, that is the role of hypothalamus. So hypothalamus is a very huge topic, okay, and also this is the most important one. But in hypothalamus, today we're going to discuss the most important question that repeatedly come in the university exam. That is the function of hypothalamus. Okay, so function of hypothalamus is the most important topic in the physiology. Rest about the structure, about the components, nuclei, this all covered in anatomy. Okay, so that is not majorly important for physiology. In physiology, we only the question only arises over the function of hypothalamus that we're going to cover in today's session. So what is hypothalamus? It is an dicephalic structure. Okay, and it is just situated below the thalamus in the ventral part of diencephalon. Okay, and it is formed by the group of many nuclei which are scattered in the walls and floor of third ventricle, and it extends from optic chiasma in the mammalian body. So many nuclei are there in hypothalamus. For example, anterior or preoptic nuclei, middle or tubular group, or posterior or mammalian group. So many are there, but here the most important is the function. So only in today's session we're going to learn about the function, the important function of the hypothalamus. So talking about the function, one by one we're going to discuss. First function we have the secretion of posterior pituitary hormones. Okay, so as we know, the hypothalamus is the site for posterior pituitary hormones, and for when we look for the posterior pituitary hormones, so majorly ADH and oxytocin are there. Okay, so oxytocin are secreted from supraoptic and paraventricular nuclei. Okay, and these hormones are transported by the means of axonic or exoplasmic flow through the fibers of the hypothalamo-hypophyseal tract of posterior pituitary. This tract name is very important to remember. Okay, then next we have uh, control of interior pituitary. Okay, so also the hypothalamus control the secretion of interior pituitary glands also by secreting releasing hormone and inhibitory hormone as well. So many are there. For example, um, GHRH, that is growth hormone releasing hormone. Then TRH, that is thyrotropic releasing hormone. Then CRH is there, that is corticotropin releasing hormone. Then next, we have gonadotropin releasing hormone. Then PIH is prolactin inhibiting hormone. So all these hormones secreted by distinct areas of hypothalamus and transported to interior pituitary. Okay. So this is all about the control of hypothalamus over the interior pituitary. Now next we have the another function. So here A, B, C, D, E. Okay. Next we have the another function, that is control of adrenal cortex. Okay, so anterior pituitary also regulate adrenal cortex by secreting ACTH, that is adenocorticotropic hormone. Okay, so this secretion in turn regulated by CRH, and which is secreted by the paraventricular nucleus of hypothalamus. Now next we have control of adrenal medulla. Right? So dorsomedial and posterior hypothalamic nuclei are excited by emotional stimuli and in turn send impulse to the adrenal medulla through sympathetic fibers and cause release of catecholamines, okay, which are sent to cope up with the emotional stress. So next we have regulation of autonomic nervous system. So also the hypothalamus have control over the autonomic nervous system and sympathetic division of ANS. As we know, the autonomic nervous system has two divisions, sympathetic and parasympathetic. So sympathetic division of ANS is regulated by posterior and lateral nuclei of hypothalamus, as well as the parasympathetic division of autonomic nervous system is controlled by anterior group of nuclei, right? Now the action of cerebral cortex on ANS are executed through hypothalamus. Then till now we have covered 5 important functions. The next we have regulation of heart rate. That is also controlled by hypothalamus. So hypothalamus regulate heart rate through vasomotor center. Okay, so please remember that regulation of heart rate will be done by vasomotor center, and that is present in the medulla oblongata. Okay, so stimulation of posterior and lateral nuclei of hypothalamus increases the heart rate, and stimulation of preoptic and interior nuclei always responsible for decreasing the heart rate. Now next we have regulation of blood pressure. Again, this is also controlled by which center? Vasomotor center. Okay, so hypothalamus regulate the blood pressure by acting on the vasomotor center, and stimulation of posterior and lateral hypothalamic nuclei increases the arterial blood pressure, whereas the stimulation of preoptic area decreases the blood pressure. Another function we have regulation of body temperature. Okay, so hypothalamus is also responsible for regulating body temperature, which sets the normal range of body temperature. The set point is, as we know in the physiological condition, it is 37 degrees Celsius. Okay. Now, next we have the another function, that is hypothalamus is also responsible for regulation of hunger and food, right? So as we know, food intake is regulated by two center. For this, we have two center. The first we have feeding center, and second we have satiety center. So the feeding center is present in the lateral hypothalamic nuclei, and the stimulation of this center is leads to uncontrolled hunger and increase food intake and resulting in obesity, and destruction of this feeding center also lead to loss of appetite, and the person will refuse to take food, right? Whereas the satiety center is present in the ventromedial nucleus of the hypothalamus, and the stimulation of the nucleus will lead to the total loss of appetite and cessation of food intake, and also the destruction of satiety center leads to hyperphagia, and the animal and the person will become obese. Okay, so this is all about the regulation of food, hunger and food, right? And the last we have the last function that we're going to discuss, that is the regulation of sleep and wakefulness. So basically, mammalian body, the present in the posterior hypothalamus, is considered as the wakefulness center, and stimulation of mammalian body cause wakefulness, and its lesion leads to sleep, and also the stimulation of anterior hypothalamus leads to sleep. So this is all about the important function, and other important function are also there, that the hypothalamus is also responsible for any kind of change in behavior and emotional changes, and also this is responsible for water balance. So other function are also there, because hypothalamus is responsible for many functions like sexual function, response, role of response to smell, then circadian rhythm. Other function are also there, but the important one are these only. And if the question over the hypothalamus will come, so the student need to explain the all the function of the hypothalamus, and these all are the important ones. Okay. Now next we have cerebellum. So cerebellum is also known as little brain, and this is responsible for coordinating movement, control posture and balance, and fine motor movement, and also this is responsible in motor learning. Okay, so cerebellum form the larger portion of hindbrain, and it coordinates the motor activity of body also by receiving sensory impulse from other part of brain and spinal cord. So again, the important function of cerebellum we're going to discuss in today's session. But in today's session, we're going to discuss these function depend by discussing the division. Okay, so based upon its function, the cerebellum is divided into three division. Okay, so first we have vestibulo cerebellum. Second we have spino cerebellum. Then third we have cortico cerebellum. Okay, so what is vestibulocerebellum? It includes floccunodular lobe that form the archi cerebellum. Okay, and talking about this spinocerebellum, so it includes lingual, central lobe, then live tuber, pyramid, vulva, many portions it includes. Okay, then talking about the last one, that is corticocerebellum. So it includes lateral portion of lobulus and ensiformis and lobular padametinus. So these all are the major division of cerebellum, and based upon these division, we're going to discuss about the function of cerebellum. So first we have vestibulocerebellum. So this regulates tone, posture, and equilibrium by receiving impulse from vestibular apparatus. Okay, and also vestibular apparatus send information regarding gravity, regarding linear movement, angular acceleration to vestibulo cerebellum through vestibulo cerebral tract. Okay, so this is all about the function of the vestibulocerebellum to regulate tone, posture, and equilibrium. Now next we have the function of spinocerebellum. So this also regulate tone, poise, and equilibrium. But here, this regulate this by receiving sensory impulse from tactile receptors. Okay, tactile receptors from proprioceptors, visual receptors, and many are there. Okay, and the spinocerebellum is the receiving area of for tactile proprioceptors, auditory, and visual impulse. Okay, and it is also known as the receiving. It is also known for the receiving the cortical impulse through pontine nuclei, and tactile and proprioceptors impulse are localized in the spinocerebellum. Now, the last we have the function of the corticocerebellum. So this area is concerned with the integration, integration and regulation of well coordinated muscular activities. Okay, and it is because of its afferent connection with the cerebral cortex through the cerebral circuit. Okay, apart from its connection with the cerebral cortex, the cerebellum also receive feedback signal from the muscle through the nerve fibers of proprioceptors. Okay, so this is all about the function of the cerebellum. Now next we have the another topic, that is the frontal lobe and parietal lobe syndrome. So first we're going to discuss about the frontal lobe syndrome. So, about frontal lobe syndrome. So, frontal lobe syndrome is a disorder that is caused by damage due to head injury. Okay. Damage due to head injury or ablation of prefrontal cortex. And various features are there of this frontal lobe syndrome. So that also we're going to discuss. That is, it will lead to emotional instability. So there is a lack of restraint and lead to hostility and also aggressiveness and lessness in the body of the patient. Then another feature is the lack of concentration that will occur, and lack of initiation and difficulty in planning any action. It will also lead to impairment of recent memory without affecting the memory or motor events. Okay, and also this is also responsible for loss of moral and social sense. Okay, and also it leads to functional abnormality like loss of control over sphincter. Sphincter here, we're talking about the urinary bladder. Okay, then there are chances of slight tremors. So this is all about the frontal lobe syndrome. Now next we have the parietal syndrome. So just a minute. So talking about the parietal lobe syndrome. Again, any kind of lesion or ablation of parietal lobe will lead to parietal lobe syndrome. And again, the features are that this will lead to contralateral disturbance of cutaneous sensations. Also, there will be loss of tactile kinesthetic sensation, and also there will be disturbance in or we can call disturbance or loss of discrimination. Okay, so this is all about the parietal lobe syndrome, and this is all about the features. Okay. Then next we have the last topic of today's session, that is decerebrate rigidity. So decerebrate rigidity is the abnormal posture changes, right, in decorticate condition, and also it involves rigid extension of lower limb that we can see, and of the lower limb and flexion of the upper limb at elbow joint across the chest, right, and wrist and fingers are also get flexed, and uh, uh, posture may develop on one side or both side of the body. So as we can see, the head and neck are arched, the arms are straight, extended, and hands are curled, and legs are also straight, and toes pointed towards the downward side. Okay. So, what about this preparation? So, whenever we talk about this preparation, so it is prepared by removing all connection of cerebral hemisphere at the level of midbrain by sectioning in between the superior colliculus and inferior colliculus. So, what is this cerebral rigidity, decerebrate rigidity? So, this is the rigid extension of all the limbs due to decerebration. Okay. So, this is rigid extension of all limbs due to deceleration. And also, arms are abducted and pronated with extension at elbow joint and flexion at wrist joint, as we can see in the picture also. And also, this decerebrate rigidity also produced by stopping the blood flow to the forebrain also. And this is done by the occlusion of the common carotid artery and the basilar artery at the center of the pons. And also the reason for decerebrate rigidity is the release of the center situated below the section from higher inhibitory control, and the inhibitory area is 4S, that is situated in the motor cortex of cerebellum. Okay, so this is all about the decerebrate rigidity, and also the short note over this topic can come. So this is also the another important topic of CNS. So in today's class, we have covered I think 6 topics in today's class we covered, that is very, very important for the CNS. And also one more class is going to occur for over the CNS, and that will be the last class. And also in that class, we're going to cover the rest important topic of CNS. And also if you have not watch the last class, that was the first class for CNS, then also you can watch that class in the recorded section. So I hope you all find the class more helpful for you. And also for more recent content, you can follow us social media handles like YouTube, Facebook, Instagram, Telegram. So thank you for today's session.